What is the bacterial growth curve with an example? Let's get start. A bacterial growth curve is a graph that shows the growth of a bacterial population over time. The curve is typically divided into 4 phases: the lag phase, the log phase, the stationary phase, and the death phase. 1. The lag phase is the initial period of time when the bacteria are adapting to their new environment. During this phase, the bacteria are not actively growing, but they are preparing to grow by synthesizing enzymes and other molecules necessary for growth. 2. The log phase, also known as the exponential phase, is the period of time when the bacteria are actively growing and reproducing. During this phase, the population of bacteria doubles at a constant rate and the number of bacteria in the culture increases rapidly. 3. The stationary phase is the point at which the growth rate of the bacteria slows down, and the number of bacteria in the culture stops increasing. This is due to depletion of nutrients, accumulation of toxic byproducts, or other environmental stressors. 4. The death phase is the period of time when the number of bacteria in the culture begins to decline. This decline is caused by the death of bacteria due to a lack of nutrients, accumulation of toxic byproducts, or other environmental stressors. An example of a bacterial growth curve is the growth of E. coli cells in a nutrient-rich medium. The lag phase would be around 1 to 2 hours, the log phase 12 to 16 hours, the stationary phase around 20 to 24 hours and death phase around 30 to 36 hours. The bacterial growth curve can be determined by measuring the optical density of the culture at regular intervals using a spectrophotometer.